Bismillahirrahmanirrahim. Yeah, we are going to talk about dosage compensation uh, as well as uh, histone variants. And before I kick off lecture, so let me tell you all that you all should go and watch Peter Becker's talk on our SSE webpage, which is about dosage compensation in Drosophila. Okay. And you should also watch, I already told you, Sandra Hicks. That's about histone variants. Okay. And when you watch these talks, you should know, I already explained you this tool, chromatin immunoprecipitation. You should know this, how to do this, and what is the use of chromatin immunoprecipitation. Okay. So dosage compensation is basically a phenomenon which is describing unequal dosage of X chromosomes in females versus males. So how genes residing on the single X chromosome in males, their dose can be equalized with two X chromosomes in females, or how the dosage of genes, which means, product of genes, transcripts, etc., and the consequent proteins, how they can be equalized in males, which carry just one X chromosome. In humans, in mammals, I mentioned earlier in this course is that one of the two X chromosomes, so there are two X chromosomes in mammals, in human females, versus 1X in males. Now, what happens that one of the two X chromosomes in mammals, in females, during very early development, it is inactivated. This shortened means, if there are two X chromosomes, so they remain fully intact to two chromosomes, but one of the two X chromosome is jumbled up, is highly condensed, and it's called bar body. And this happens by activation of a large non-coding RNA, which is nearly 17,000 nucleotide long, non-coding RNA, which is called ZIST. ZIST binds to the X chromosome. Let's say this one is to be inactivated. ZIST will bind and attract all the chromatin or gene silencing machinery, which also includes DNA methylation, methyltransferases, histone methyltransferases, the methyl mark binding proteins like PRC1 complex, this is PRC2 complex, and everything is kept as a silent chromosome. Complete chromosome is silenced, and this is also facultative heterochromatin because these two X chromosomes during earlier part, initial few cell divisions, they both are being transcribed, they both are euchromatic. But then there comes a stage when one of the two X chromosome is randomly silenced. And when I say randomly, let's say you are going from single fertilized egg to two cells to 4 cells. You keep dividing like this mitotically, and then you have few hundred cells. In these 100 cells or so, the choice which of the two X chromosome is going to be inactive is random. It can be this one in one cell, in another cell, this can be silenced. But once it is converted into bar body, mitotically when these cells will pass on the inactive chromosome, they will then keep the one which is selected, mitotic inheritance. So you see the machinery for heterochromatin, which is DNA methyltransferases, polycomb complexes, etc. They are performing evolutionary conserved function in humans as well. Then comes the invertebrates, flies, C. elegans worms, etc. There also, we know males contain XY and females contain 2X chromosomes. However, in contrast to the mammals, instead of inactivating one of the 2X chromosomes, the 2X chromosomes in females, they remain normal. Their transcription and their gene regulation remains normal. No inactivation takes place there. However, in males, in male flies, this color, light to dark, is shown, is explaining that the genes residing on the male X chromosome, all of them, their transcription is enhanced. Transcription regulation goes twice as much of single X to equalize the dosage of genes on 2X in the females. This is shown here. 2 females and single X is producing as much of gene products as 2X will generate. In C. elegans, there is equal repression, so they are reduced by 50%. In both the X chromosomes are reduced by 50% to equalize the dosage of single X. So is it clear what is the difference between X inactivation and dosage compensation?
Yes, sir.
Now, if you pay attention, these are the autosomes. These are the large, giant salivary gland chromosomes, which are also called polytene chromosomes in flies, polytene chromosomes. So these polytene chromosomes, these are all the ones which are visible largely as blue. They are the autosomes, and the single X is this one, which is immunostained with a protein which we'll soon see, which is a histone acetyltransferase, which binds to X chromosome and hyperactivate genes residing on single X. This can also be observed by simply observing the phenotype of eye color. Since the X chromosome contains white pigmentation gene, which is responsible for giving red eyes in flies. So white resides on the X, we already know. If you have 2 X chromosomes in females, eye color looks like this, and the eye color of males, which contains single X, also looks similar, which means white gene residing here on the X chromosome, single X, is hyperactivated, and we have equal dosage of white pigmentation gene, which results in normal eye color. If we, in flies, if we remove 1X, then flies with single copy of white gene, this is how the eye color should look like. And this is how it looks like if you have single copy in females. Since there is no hyperactivation of the gene here on single X, you don't see single X giving eye color like this, which happens with the single X in the males. So this is a phenotypic observation of X-linked dosage compensation in fruit fly males. And if you add an extra copy in fly males, the eye color becomes more than the normal eye color in males and in females, because if you have added an extra copy of white, which is a transgene, let's say, which is in addition to the wild type X chromosome copy, this shows you have hyperactivated white here and 2 transgene copies even on the autosomes. Now, how it happens, we'll soon see. It somehow is linked to the process of sex determination as well. How, early embryo knows that I have 2 X chromosomes or I have single X chromosome. How it decides single X chromosome or 2 X chromosomes, why it does not hyperactivate these two during very early stage embryonic development, and how it detects that I have a single X, so that all the genes residing on the single X, they need to be hyperactivated. So all this happens during sex determination at early embryonic development, when embryo realizes that I have single X or 2 X chromosomes. X to autosome ratio we learned in our earlier lectures, that if X to autosome ratio in fruit flies is 1, you develop as female. If X to autosome ratio is 0.5, you develop as males. But how actually at molecular level it happens? So there is a gene called sex lethal. All these genes, sex lethal, male sex lethal, double sex, etc., all these were discovered in genetic screen in fruit flies, for which showed X-linked sex lethal phenotypes. So sex lethal, it encodes a female specific RNA binding protein. In this RNA binding protein, it actually regulates the splicing and translation of key messenger RNA, which are involved in sex determination and dosage compensation. So this sex lethal, it is present in males as well as females. This gene is present in both males as well as in females. However, the splice variant, which is produced in females, is functional, and the splice variant in males is non-functional due to incorrect splicing. So what we have just learned, in males, sex lethal is off, or let's say non-functional, because the messenger RNA is incorrectly spliced, and sex lethal is non-functional. In case of females, sex lethal is correctly spliced, and the protein product of sex lethal, it regulates the splicing and translation of key messenger RNAs, which are involved in dosage compensation as well as in determination of sex in flies. Now, this is what is happening. So in early stage embryo in fruit flies, at which is like 12 to 14 hours. In females, X to autosome ratio turns out to be 1, and there's a burst of sex lethal transcription. This is the sex lethal gene, which is present in males and females. But its burst of this transcription is kicked off or initiated from early promoter. And later in development, this transcript of sex lethal is maintained by maintenance promoter, which is this one. So sex lethal functional messenger RNA is made, sex lethal protein is made. This sex lethal protein is functional, and then it contributes to, we will soon see what it does. However, if this sex lethal transcription kicks off later in development, so here, early development, the early promoter, this arrow tells you transcription of sex lethal started early during development. However, if you fail to activate during early development, the maintenance promoter, which is called the late promoter, maintenance promoter, the transcript which will be generated by transcription from this promoter, that will be non-functional messenger RNA, because it will be incorrectly spliced, and the protein from the functional messenger RNA, which was the sex lethal protein, it then goes and correctly splices this. So it's self-regulating itself as well. And this correct splicing then leads to functional messenger RNA, and translation takes place, and you have sex lethal protein continuously being made from early to later development. Now, what this sex lethal protein does, it blocks the translation of messenger RNA of a key gene which is called male sex lethal, MSL2. Male sex lethal, if translation of male sex lethal is blocked, then you have only females being made. And this is happening early during development when you have sex lethal transcript, early during development is correctly spliced. From this early promoter transcript is generated, which is maintained by maintenance promoter late during development. And what happens that this blocks a key male specific gene, which is MSL2, and its translation is blocked, and you have female sex is determined. This is what is happening in males and females. So you have females, X to autosome ratio is 1, sex lethal is transcribed. This sex lethal then also contributes to correct splicing and eventual translation of another key sex determination gene, which is called transformer. So you have TRA and TRA2 being made, which together play a role in female specific isoform of double sex, and female sex differentiation takes place. At the same time, sex lethal also blocks the translation of MSL2 which I told you here. And this absence of MSL2 means no dosage compensation, because MSL2 is key component of hyperactivation of genes on the X chromosomes. In contrast, in the males where you have single X, which is like X naught means no second X, XY, ratio is 0.5. Now, incorrect or non-functional transcript of sex lethal is formed. This cannot contribute to the splicing, or since sex lethal is not present. So now transformer is absent, only transformer 2 is present, which leads to male specific isoform of double sex, and male sex differentiation takes place. At the same time, absence of sex lethal leads to translation of MSL2, which leads to hyperactivation of all the genes on X chromosomes in male, and you have dosage compensation. If you make mosaics, you remember the flip FRT clones you can make in somatic clones we talked about in flies. If you can generate sex lethal mosaic cells, so cells which lack sex lethal, they will contain male specific components of dosage compensation. So let's say you have a cell which was heterozygous for sex lethal mutation. This is the sex lethal wild type copy. This is, you generated mitotic clones, and after mitotic clones, you have now generated sex lethal minus minus, and they become wild type, sex lethal homozygous wild type. This will be minus minus all the cells, and they will be plus the wild type. This is called chimera. Tissue is chimera, which contain, you know, two different genotypes. Now, in the absence of sex lethal in progeny here, you will have all the components of dosage compensation, DCC complex, in progeny of these cells, which will be absent here. And you can, in real time, in tissues, you can observe this phenomenon. Now, what are the components of dosage compensation? All this is very nicely explained, and you should watch, I told you already, Peter Becker's talk. So in screen, what they found, MSL1, MSL2, MSL3, MSL4, male sex lethal 1, male sex lethal 2. So when they were absent in males, they were showing male specific lethality. So mutant males, they don't develop, but females are unaffected. So females were normal. And MSL2 was truly a male specific. In females, MSL1 to 3, they exist. MSL1 exists, 3 exists, 4, but 2 is really male specific. All the MSLs, 1, 2, 3, 4, all of them, they associate with X chromosome in males, but not in females. This is what I showed you in the earlier. This one is MSL2, I believe, staining. Now, the dosage compensation complex, they also contain a non-coding RNA, which is called ROX RNA. So look at the X chromosome regulating complexes, or whatever you name it. In human female, we talked about ZIST, non-coding RNA. which binds to a future inactive X chromosome and, you know, turns it off. In case of flies, the ROX RNA acts as structural component of the DCC complex, and then they bind to chromatin. We'll see what is role of ROX RNA in bringing this whole complex. MOF is a histone acetyltransferase, which also plays a crucial role in dosage compensation in males, and MOF actually hyperacetylates histone H4 lysine 16 acetylation is the covalent modification which goes hand in hand with dosage compensation in fruit flies. Oh, I was wrong. This was not MSL2, this is MOF, which is acetyltransferase, H4 lysine 16 acetyltransferase. Okay, now how the dosage compensation complex, it learns that I have to go and bind to only X chromosome in males and not on autosomes. So what they have discovered that there are two non-coding RNAs which are critical for dosage compensation in fruit fly, ROX1 and ROX2. And both ROX1 and ROX2, they are only found, their transcripts are only found in males. And the non-coding RNAs, they actually lead to targeted distribution of dosage compensation on X chromosome. For example, the ROX2 locus 10C is the cytological location of ROX2, and the ROX1 is 3F. They are present, their genetic locus which generates ROX1 and ROX2, they are present on X chromosome. But they are only present, the transcripts are only present in males. Their transcription somehow provides early targeting of MSL2, MLE, MSL2 targeting onto X chromatin. And through RNA fish onto polytene chromosomes, they have found that there are nearly 35 sites which contain ROX2, ROX1 RNA plus MSL2 onto chromatin. So these are the loci where transcription takes place and ROX transcripts are generated. But there are 35 or more than nearly 35 sites during early development which contain ROX1, ROX2 plus MSL2 bound onto chromatin. And from here, these early sites, then this complex spreads to other sites in the rest of the X chromosome. And we call them cis spreading sites. So the early targeting site, and then along the same arm of chromosome, they spread. They have also shown that if you make transgenic flies in which you ectopically incorporate ROX gene, which is going to generate transcript of ROX on autosome, it also on the autosome will recruit MSL complex, but this transgene can also go to the X and bring dosage compensation complex, which means ROX is crucial for targeting the MSL complex or dosage compensation complex onto chromatin, because on the autosome it does the same thing. These are the sites which provide targeting of, so for example, this is ROX transcript being generated. It brings in, it helps in assembly of dosage compensation complex or MSL complex in males. And then it spreads first to initial sites and then, to more distant sites, nearly 35 such sites. But there are high affinity sites where initial targeting takes place, for example, and then from high affinity you go to low affinity site and eventually to the lowest affinity site, and all the X chromosome is labeled or is bound by uh, spreading of ROX RNAs and targeting of consequent targeting of MSL complex. As we learned that uh, this phenomenon is happening in males only, so there is a difference in the chromatin structure of male and female X chromosome. Uh, Female polydine, this one, uh, you have two X chromosomes there, both homolog. They are aligned side by side as a single X. So on polydines, you won't see two X chromosomes. They will be aligned side by side. They will appear as single X. And the size of single X, it's like in males, is equivalent to the size of 2X in females. You also have histone H4 lysine 16. You can easily recognize lysine 16 if you can stain with MOF acetyltransferase. You can easily see male polydines. There are chromatin remodeling proteins as well. The ATPase chromatin remodeling proteins which contain ATPase domains. Imitation switch is one such protein which is shown that if you have hypomorphic mutation in imitation switch, so I switch mutation, the structural organization of the X chromosome in males is affected. You see, structure of X chromosome is totally collapsed because it is involved in maintaining the association of dosage compensation complex on X chromosome in males. So before I move on to the next topic, I would like to go back to human females, where we learned that ZIST, the non-coding RNA, it gets transcribed from the X chromosome. Let's say I means the future inactive X chromosome. So ZIST then is 17,000 nucleotide inactive X chromosome. It binds and it recruits the DNA methyltransferase machinery, histone lysine methyltransferases, which are PRC, 2 complex, and eventually PRC 1 complex, which contains polycomb, and this whole X is converted into bar body. In 1959 or 60, this phenomenon was discovered by scientists, and this bar is honoring his name, Bar Body, B-A-R-R. How the other X chromosome, which is to stay active, remains active. Why you have, once you have zest, why it doesn't go and binds to active X chromosome as well? This chromosome, there is also a long non-coding RNA, and this long non-coding RNA is called T6. This T6 blocks binding of zest onto this chromosome, and all the genes on this chromosome, they are protected from being inactivated by, process of X inactivation in females, in humans. Role of non-coding RNA. This is one example, long non-coding RNA. There are siRNAs as well, which are dicer and other endonucleases, which specifically target double-stranded RNA and convert them to 21 to 23 nucleotide long siRNAs. There are microRNAs as well. And now they have pRNAs as well, which are in gametogenesis. So there are many classes of non-coding RNAs. But what you will learn that these non-coding RNAs, they also act as signal for recruiting DNA methyltransferase in plants and mammals. DNA methyltransferase or histone lysine methyltransferases as well. So non-coding RNAs, they not only result in post-transcriptional gene silencing by, targeting the messenger RNA and converting it into double-stranded RNA, which is then targeted by dicer. So the messenger RNA of a gene is degraded, and we call this post-transcriptional gene silencing because gene is transcribing functional messenger RNA, but it is being targeted by siRNAs or microRNAs. At the same time, siRNAs are also shown that they can recruit silencing machinery transcriptionally, which we call TGS, transcriptional gene silencing machinery, onto promoter elements and cause DNA methylation or histone methylation, DNA or histone methyltransferases, and then lead to silencing. So Dr. Afzal is going to talk in detail about these phenomena. Let's talk about the histone variants now. And you will find beautiful data and how histone variants, they act onto chromatin in Sandra Haig's talk we had, and I think some of you already attended, but I would encourage you to go and watch these talks again and again to get a clear understanding of these fundamental principles in chromatin biology. So histone genes, as we learned already, they are the integral components of the chromosomes, and they are responsible for correct packaging of DNA in the form of chromosomes and eventually packaging in nucleus. Histone genes are normally present in cluster. They are present in cluster like this. And they don't contain introns. The And then there are, these genes which are more abundant, which are prevalent. Most of the chromosome is packaged in these histone H2A, H2B, H3, and H4. They appear as cluster. We call these histones as canonical histones. Canonical histones. These canonical histones are the ones which mostly cell utilizes to package DNA in nucleosomal form. And as one expects, their expression is maximum during the S phase, and only residual levels are present in rest of the cell cycle. So when you have cell cycle, you know G1, then S phase, then you have the G2, eventually M phase, etc. S phase is the time where you have DNA replication is taking place. And during this DNA replication, when newly assembled DNA is to be packaged in chromatin form, this is where you have highest level of transcription of histones taking place. In rest of the, after mitosis, in rest of the cell cycle, you have only residual levels. However, during interphase also, when you have DNA damage and cells need to repair DNA, damaged DNA, you have transcription of histones as well, other than S phase. In addition to canonical histones, you have variants of H3, H2A, H4, etc. Not H4, H2, H2B, H3. Other than H4, uh, all the histones, they encode variants. Variant means they will be, in some cases, they are 100%, not 100%. In some cases, they are entirely similar, identical, except few amino acids. And in some cases, they have significant variation than canonical histones. As I said, so there are subtle variation as well as large scale primary structure can also be different because additional amino acids are present. As I said, you have maximum transcription of histones is taking place during DNA replication during S phase, which we call the replication coupled incorporation of the histones. And in animals, this replication coupled histones, they are recognizable by presence of 26 base pair stem loop in messenger RNA of these sequences. Plant histones, they possess long poly A tails, and there are no such stem loop which is present in replication coupled histones in animals. In the S phase, there is a specialized complex which we call chromatin assembly factor or chromatin assembly complex. When DNA replication takes place, you can see the newly replicated DNA is being reassembled in the form of histones. And if you pay attention to colors, so these are shown that the mother cell or the mother's parental histones, they are not degraded, they are equally distributed to newly replicated DNA. So these ones are, for example, old nucleosomes, but there are new ones as well, these light colored. Okay. And all this is done by chromatin assembly factor CAF1 together with this PCNA, proliferating cell nuclear antigen, all of, so the DNA polymerase, this is replicating newly, this is involved in replication of DNA, CAF complex together with PCNA, CAF consists of 3 subunits, and they interact with pCNA so that coupling of DNA replication together with chromatin assembly is ensured. And if you have mutation in CAF subunits or CAF1, it results in defective inheritance, epigenetic inheritance, because chromatin is not properly assembled, and the chromatin state, let's say genes were off here, if CAF1 mutation is there in daughter cells, eventually in daughter cells, those genes which were epigenetically silenced, they are not epigenetically inherited in proper chromatin conformation, they will be switched on. This is what I explained you that, there's a model for epigenetic inheritance that newly replicated DNA is assembled into histones or nucleosomes which contain mother's nucleosomes, which parental nucleosomes, which are passed on equally, as well as newly replicated histones. And idea is that these nucleosomes, they act as signals, and then epigenetic states are also restored. At the same stage here, in plants and in mammals, I told you DNA methylation machinery, the maintenance methyltransferase, which was DNMT1, or in Arabidopsis it was MET1, it comes in right here in the replication fold. It recognizes the hemimethylated DNA like this and restores the methylated state. And this is what we call epigenetic inheritance. This is epigenetic state is being inherited. Now let's look at the variants. What function variants has in the chromatin organization, chromatin structure, and gene regulation, etc. So canonical histone looks like, you have N and the C termini. The canonical H2A is normally present in, it's abundant in chromatin, but there are 2 variants as well. H2AZ, H2AZ variant, there are 15 amino acid substitution in the N and the C terminal domains. So there are these 15 amino acids in the N, which are sprinkled 2, 3, 4, 5, 6, then in the core, and 1 in the C termini. Another variant of histone H2A is called macro H2A, and it contains 240 additional amino acids other than this canonical histone plus these variations. There are H2A specific variations from canonical histones in the N as well as in the C region, but 240 additional amino acids. Now, what macro H2A does, it's reported that macro H2A, it specifically associates with inactive X in humans. The inactive X, which is due to the zest, so inactive X will get macro H2A incorporated into inactive X. The H2AZ is still a bit confusing. It's reported both in silencing, the heterochromatic silencing, kind of facultative, whatever, as well as it's also present in euchromatin. However, there's also data in the yeast which suggests that, if you have, if from here to here somehow heterochromatin exists, and from here to here euchromatin exists, H2A Z may act as boundary between heterochromatin and euchromatin, and that's why when it is disturbed, we see euchromatin being disturbed or heterochromatin being disturbed. But if you look at, I think, Sandra's talk, you will get much interesting idea listening to his data. Then we have H2AX, which is another H2A variant. It has only 4 amino acid difference. There's only 4 amino acids. So here in the C terminal, you will see just 4 amino acids. And the serine in this is phosphorylated when DNA double strand break happens. So this H2AX is somehow DNA damage or DNA repair specific histone. When DNA damage will take place, it will be incorporated into H2AX, and then the DNA repair machinery will be, all the replication or whatever other processes will come to a halt and repair will start. H3. H3 is very interesting. There are 4 different variants. You have 3.2. So besides canonical H3, you have 3.2, 3.3, SEN-PA, and SEN-H3. H3.2, it differs by a single amino acid. There is serine to cysteine in C terminal. Look, just single amino acid. How, think of difficulty in studying H3.2. You have to have monoclonal antibody maybe, or some tagging of this gene with some other tags, but then the problem will be how you ensure this is properly functioning in this tagged form. This 3.2 is only synthesized during the S phase of the cell cycle. 3.3 is very interesting. Steve Henikoff's group in Seattle at Fred H. Henson Institute, and the guy who discovered working with Steve Henikoff is called Kami Ahmed. He grew up in Canada. He's a Pakistani. He is the first author of those 3-4 papers on H3.3, which were published by Steve. So 3.3 is actually variant by 4 amino acids and position 87 to 90. Position 87 to 90 amino acids in histone H3, they are different in H3.3. What they discovered that 3.3, it localizes only to the active euchromatin. Wherever gene transcription, wherever genes are on, we have H3.3 there. And another interesting thing they showed, that it is assembled into histones. It is assembled into nucleosomes, both in replication coupled and replication independent manner, which means during S phase and wherever transcription is taking place, which mostly takes place outside the S phase in the non-dividing cells, H3, you will find H3.3. And these 4 variant amino acids, they prevent canonical H3 from replication independent deposition. Did you understand? So H3.3 and canonical H3, they differ in those 4 amino acids. And these 4 amino acids, they enable H3.3 to be assembled into nucleosomes in replication independent manner as well. But these 4 amino acids which are absent in canonical histone, due to that, this H3.3 cannot be assembled in replication independent manner, and H3.3 is only assembled into nucleosome or chromatin in replication coupled manner. H3.3 also, when now the question you should ask is, let me add a slide here. Can you see my slide still? Do you see my screen?
Yes, sir.
Yeah. So question arises, let's say a gene is off. And now there is a signal that this gene should be switched on. All the machinery which needs to turn on gene, they will get assembled, RNA pol 2 is there, and they will slide through coding region. This is the promoter region, for example. This is transcription initiation complex. When it will move through gene body, and we call this transcription elongation phase, RNA pol 2 gets serine 5 of RNA pol 2 in these heptide repeats. Sorry, serine 2 in these heptide repeats, it gets phosphorylated and RNA pol 2 is moving. Now here, the nucleosomes, the same gene when it was in the off state, it contained canonical histone H3. When transcription takes place, you should know that all these as the RNA pol moves through RNA pol 2, so this is the C-terminal domain of RNA pol 2, let's say. When it moves, RNA pol 2 moves through the gene body, the nucleosomes ahead of RNA pol 2, they are evicted. H3, so these nucleosomes, they are removed, they're disassembled. If I remember correctly, H3, H4 tetramer is removed, H2, H2B still can stay there. And when RNA pol 2 passes through this gene body, behind the RNA pol 2, you have nucleosome reassembly takes place. And here you have H3.3 replacing the canonical H3. Now when gene from off to on takes place, the nucleosomes, they contain, instead of canonical H3, they contain H3.3. It replaces the H3 at RNA pol 2 in one transcribed loci. And I told you how in earlier lectures we talked about how genes from off to on states are converted. We talked about, cell signaling, serine 10 phosphorylation, or serine 28 acetylation, um, all this chromatin targeted cell signaling, or let's say we also talked about histone ubiquitination. You should also think of, let's say, facultative heterochromatin, which is, I make this one PRC2, which is EZ, EED, etc., which convert H3K27 into trimethylation, recognized by PRC1, which has H2A ubiquit, H2B. Najma, H2, why I'm forgetting?
Sir, H2AK119.
H2A119.
Ubiquitination.
Ubiquitination and polycomb in PRC1 recognize the trimethylated state. So you should think about how this silent state can be switched on. There has to be cell signaling events, which then will lead to reversal of epigenetic marks of K27 acetylation or these ubiquitination marks, etc., replaced by trithorax group associated active marks and activation of the gene and eventually H3.3. This is how you should think of more dynamic and holistic picture when you talk about chromatin. Don't just look at binary strains. Other histone variant, this is the last slide, I believe. Yeah. So is the of histone H3 is SNP-A. And SNP-A is actually centromere specific. Look, this is the staining of antibody against SNP-A. And look, these are centromere specific histones, which means the centromeric DNA is, when it is chromatinized into nucleosomal form, it does not contain canonical H3, but it contains SNP-A. And these are specialized nucleosomes. These are specialized chromatin because they then result in assembly of kinetochore complex, where kinetochore, the mm,
Microtubules.
proteins which bind or recognize these specialized histones, which contain SNP-A, SNP-B, etc. They are recognized by specialized proteins which are cohesins, no, not cohesins.
सर एक सेपरेस होती है.
Separase is involved in separation. Cohesin is involved in, keeping sister chromatids together. Separase is the one which separates the sister chromatids. Anyway, there is a specialized protein complex as well, which recognize
APC, anaphase
such SNP-A containing nucleosomes, and the microtubules then pull sister chromatids apart. So this is the role of H3 variants, SNP-A, SNP-B in correct segregation of chromosomes during mitosis. SNP-A, it remains associated with the centromere during transition from histones to protamines during spermatogenesis as well. Now, this process of gametogenesis in males, in spermatogenesis, most of the histones are replaced by specialized proteins called protamines, but the SNP-A, it remains associated with chromosomes, and they, after fertilization, you can identify male genome by simply staining them with anti-SNP-A. Also, there is SNH, which is also centromeric histone, you can see here. So they are SNP-A, SNP-B, SNH. They are H3 variants in chromatin. So last lecture, so today is 14th lecture, but I'll take one more lecture, which will be Wednesday. Wednesday will be my last lecture. And we'll talk about, epigenetic cell memory, which is basically all about what we are doing in our lab at LUMS using Fly. And you will also see how we, use different functional genomics tools, for example, RNAi or, genetic tools, etc., to study chromatin and gene function in fruit flies. Any questions? Don't miss
सर एक
this
क्वेश्चन
lecture.
सर एक क्वेश्चन है
जी
सर, ये जो अभी आपने सेन पी और सेन पी ए और सेन पी बी का बताया,
या
तो सर ये क्रोमोसोम स्पेसिफिक होते हैं?
या all
सर ये
all
क्रोमोसोम
these
स्पेसिफिक होते हैं?
all these variants are chromosome what do you mean by chromosome specific?
सर आपने कहा है कि हम इसकी एंटी प्रोटीन बना के चेक कर सकते हैं कि ये इसकी मतलब कर सकते हैं, हम इसकी प्रेसिपिटेशन चेक कर सकते हैं इसको। तो सर
अगर एंटीबॉडी बनाएंगे
तो ये हर क्रोमोसोम की अलग होगी, हम आइडेंटिफाई कर सकते हैं कि कौन से क्रोमोसोम पे है?
So, Taha, these CEN-P are the variants which are associated with centromere. That's why they are called CEN. CEN for the centromere.
नहीं, let me understand his question.
वो तो ठीक है, सेंट्रोमियर पे है, पर क्या हम इसे क्रोमोसोम को आइडेंटिफाई कर सकते हैं कि कौन से नंबर का है या कहाँ कौन सा वाला है?
No, 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 no. Because you will have CEN-PA on all the centromeres.
ओके सर, ओके। मैं यही पूछ रहा था कि वो आइडेंटिटी के लिए काम नहीं कर सकता।
No, no, no.
इससे हम सिर्फ सेल साइकिल का अंदाजा लगा सकते हैं।
नहीं, they are present even outside the cell cycle as well. Centromeres are a centromeric DNA is assembled into specialized nucleosomes which contain CEN-PA.
सर मुझे इसकी एक दफा रीजन बता दें कि हम इसको क्यों स्टेन करते हैं?
हम इसको क्यों स्टेन करते हैं?
यस सर, इससे हमें क्या पता चलता है?
Let's say you are studying assembly of chromosome into nucleosomes, and you found H2A variants. You discover H2A variants, and that gives you an idea that okay, all the nucleosomes, all the chromosomes, they do not contain just H2A. There are, these kind of variants, which means a specific variant in specific position is impacting chromatin and gene regulation. Now, you say, is these variants, they are only present in H2A, or sorry, H2? Are there variants in H3? So you go on hunt for histone H3 variants, and you find H3, 3.2, etc. But then you also find some proteins which are named here CEN-PA, CEN-PB, etc. You have no idea they are variants of H3 or no. You simply start doing all analysis. You generate antibody against H3.3. You generate antibody against CEN-PA, CEN-PB, H, and then you stain nucleosomes, you stain chromosomes, and what you discovered that H3, its antibody is staining all the nucleosomes which are in the euchromatin. And CEN-PA is going to centromeric heterochromatin, or in mitotic chromosomes, when you look at, you see CEN-PA, CEN-PB, etc. only coming here. So that's why you do these trainings, to understand, to study. So from then you say, okay, CEN-PA, CEN-PB can be used as excellent mitotic marker as well. Clear?
यस सर, ये लास्ट लाइन में सर आपने बता दी रीजन।
Whenever you discover a new gene, you have to characterize that. ये मैंने बड़ी डिटेल में एक पूरा लेक्चर दिया था.
यस सर।
Any other questions? So if there are no more questions, let me say goodbye, but let me also reiterate that make sure you read all the papers and watch the talks. I recommend it. Okay? And don't miss next lecture. Allah Hafiz.